Spliced HLA-bound peptides, a black swan event in immunology. It was historically believed that all swans are white because these were the only ones that had been seen. However, in 1697, a Dutch explorer unexpectedly discovered black swans in Australia. The black swan theory is now a metaphor that describes an event that comes as a surprise, has a significant effect, and is often poorly rationalized. In antigen processing presentation pathway, a protease cleaves a linearized protein to generate short immunogenic peptides, which then dock onto the MHC molecules. T cells learn how not to react to self-epitopes. However, even a point mutation can change a self-non-immunogenic peptide to a highly immunogenic neoepitope. Another very controversial means of generating neoepitopes is peptide splicing, which until very recently was considered a scarce event. Sometimes, those proteases catalyze the reverse reaction, the ligation, as well. These cleave-ligated, non-genomically templated immunogenic peptides are called spliced epitopes. As late as 2016, the number of known spliced epitopes could be counted on one's fingers, but it wasn't even remotely close to reality. Later on, two teams, including ours, showed that the splice sequences account for almost 1/3 of the HLA-1 immunopeptidome. So, the large-scale finding of MHC-bound peptides that are not linearly templated in the genome is a black swan event indeed. In this review paper, you will find the most recent debates on spliced peptides. You also get familiar with the rules governing peptide splicing and get to know some of the challenges in finding spliced peptides in the immunopeptidomics data. Check out Spliced HLA-bound peptides, a black swan event in immunology.